Okay, so this is going to be part 2 of the viruses and cancer recorded lecture. So after watching the YouTube video, just in summary, characteristics of cancer cells, a lot of genetic changes, increased telomerase activity. The cells become immortalized, meaning they don't die when they're supposed to. They grow faster. Lack of contact inhibition. This is definitely one that you see in a plate. Normally, cells grow until they're confluent, so all the cells are touching each other, and then they know we don't have any more space, and they stop growing. Lack of contact inhibition means, you got a nice even layer of cells, and then they just start balling up on top of each other. Anchorage independent, so they're not dependent upon sticking to each other anymore. Or you can take cancer cells and inject them into a semi-solid medium and they'll grow. They don't necessarily need as many nutrients. The big one here, loss of cell cycle control. They keep dividing. Some of them have changes in structure, and many of them produce specific antigens on their cell surface. Now, this is something that often the immune system can target. But if you evade the immune system, then you can become a successful cancer cell. But our immune system is primed to take out some of these cancers. Also, the fact that there are specific tumor-associated antigens means these might be targets for therapy. And we'll talk more about that later. So remember, cancer is a multi-step process, and the book really emphasizes this. The video emphasizes this. This is a multi-step process. So oftentimes you get one mutation in a cell, and then before it can become a cancerous cell, either it kills itself or your immune system takes it out. But sometimes cells go on to become cancer cells, right? And these are able to avoid cell death, circumvent the need for other growth signals so they can grow independently, avoid the immune system. This is important, right? If you're going to become a mass, a tumor, you've got to be able to get your own blood supply. They might be able to metastasize and spread to other areas. And tumor suppressor genes lose their function. A lot of people think about you're stimulating growth signals, and that's often the case. But if you remove growth suppression, that can also lead to cancer. Now, a lot of research has been focused on how retroviruses or viruses can cause cancer. And if we look at your typical retrovirus, right, you don't have necessarily all that many genes. You've got these repeats at the end of the genome. You've got some control regions, promoter regions here. The GAG gene, so this is encoding matrix proteins and core proteins that are going to help form the structure of the virion pole. This is going to be a kind of a multifunctional gene or a holoenzyme that's going to include your reverse transcriptase, your RNase, helicase, integrase. Envelope, this is going to be your spike proteins, your glycoproteins here. Now, not all retroviruses cause cancer. Some of them do, some of them don't. Those that do cause cancer often have a viral oncogene. Now, in the case of Ralph sarcoma virus, the first one that was characterized, you have this SRC gene. And this gene is not a structural component of the virus. It's not required for replication. So it's not going to be found in the virus itself. This would be expressed during infection. And this gene, actually, in Ralph sarcoma virus, this is a cellular analog of a tyrosine kinase. Now, this is important. This gene was taken up or stolen by mistake from the host cell, and then it acquired some mutations and it became a viral oncogene. So this tyrosine kinase is able to promote cell growth. And so if the virus has this, in this case, SARC, this viral oncogene, then it can induce cancer. Now, some viruses induce cancer through insertion or inducing rearrangement of the host genome. Now, retroviruses, where they insert is important. If they insert into, say, a tumor suppressor gene, then they could cause the inactivation of that tumor suppressor gene. Or, depending on where they insert, you could get constitutive expression of this viral oncogene. So this just gets turned on all the time. Or the virus could insert itself so that it stimulates cellular growth signals. So depending on where the virus inserts, it can cause cancer in a couple different ways. So the virus, remember, first we have to bind to a receptor on the cell. Here we've got membrane fusion, so this would be entry, uncoding. Remember, these are RNA viruses, so you need a reverse transcriptase to make DNA. The viral DNA is then going to traffic to the nucleus, and then it needs the gene integrase to integrate into the host genome. And so in some situations, in some cases, this can be one holoenzyme that can have multiple functions, or you can have multiple enzymes doing these functions. Now, looking specifically at viral oncogenes. Right, so this figure from your textbook shows a couple different ways that can lead to cancer. Right, so in the first situation on the left here, we've got a viral oncogene that has, or a viral insertion sequence that has inserted itself upstream of a proto-oncogene, so a cellular gene that can upregulate cell growth. Normally, it would only be activated at certain times or maybe in low amounts, but if you've got a virus inserting upstream of that into the control region, now you get excessive expression of this proto-oncogene, and you get lots more protein products than you're supposed to do. Maybe these are transcription factors, or maybe these are kinases, and you lead to abnormal or uncontrolled cell growth. There's several mechanisms that your book goes into. On the other side of this figure, we have one more. And so here we have a viral oncogene. It gets inserted into the host DNA. And when the viral oncogene gets expressed, it is likely, maybe it's a homolog, or it's similar to a cellular proto-oncogene. And when this gets produced, it can lead to cancer by making unusually active proteins. So maybe you get a protein that's constitutively active, and that can lead to the cell getting more growth signals than it's supposed to have and lead to cancer. So what can those constitutively active cells do? How can they cause problems? So normally, right, you've got a pretty complex mechanism in place where you've got receptors on the surface, and they pick up growth factor signals from other cells in your body, your endocrine system or other hormones, and they send out growth factors. So when it's appropriate, and if the cell is supposed to grow or respond to that growth factor, it's got a receptor for it. And on the intracellular part of that protein, you've got this tyrosine kinase that can then activate another protein that can then travel to the nucleus, upregulate, or act as a transcription factor, alter the transcription regime of the cell and lead to growth. Now, a oncogene or a viral oncogene, perhaps, this tyrosine kinase could be constitutively active or always active. So you're no longer dependent upon growth factors from other cells. You're no longer dependent upon signals from your body. You're always active, and so you're telling the cell that it constantly needs to grow and divide. You can also have internal growth signals too, and if those proteins are no longer dependent upon their receptors, or maybe you've got a protein that normally you need something to bind to this protein to activate it, and maybe you get a mutation in either this receptor or this growth factor, this internal growth factor and this internal receptor. And normally, say these two proteins are supposed to come together, signal, and then separate. But maybe you've got a mutation in one of these factors, and once they bind, they get permanently glued together and they can no longer separate. Now it's constantly or constitutively triggering the growth. So there's a lot of different ways that cancer can form, and viral oncogenes can work through these mechanisms and more. Endogenous retroviruses. These are kind of cool because they make up a really significant portion of our genome. Human endogenous means, they're part of a certain side of us already, retroviruses. So our ancestors, many, many, many moons ago, became infected by a retrovirus, and then over time it picked up mutations, and now those viruses are no longer able to form infectious virus particles, and they're kind of stuck or stranded in our genome, and they can't really make real viruses, and they can't continue their infection cycle. So we've got a large percent of our genome, more than 8% of our genome, is just kind of these stranded, half-dead endogenous retroviruses. And we've talked about this, I think, before, is that some of these endogenous retroviruses produce proteins on occasion, and some of those are very useful, like in the formation of the placenta. Now, some of these viruses, we don't know if they do anything at all. Some of them make proteins, we don't know what they do. There is a possibility, and I want to really emphasize that this is only a possibility. All right, this is a big maybe. There is preliminary evidence that maybe some of these endogenous retroviruses may be involved in cancer, autoimmune diseases, and some neurological conditions like schizophrenia. It's possible, again, maybe, that some of these endogenous retroviruses can be triggered when you get another viral infection, and can maybe be associated with disease. But this is still very preliminary. All right, we're going to stop this lecture here for part 2, and part 3 will follow.